Hi, good morning, everyone. So today I'm going to share some of our results in the biomarker development in aging. So our company is interested in develop new treatment for age-related disease. Specifically, we are applying generative adversarial network to de novo generate new compounds to treat age-related diseases. And another part of work we are interested in is in the age research. So aging research, as we believe aging is one of the key components to contribute to the age-related disease. If we know better of the aging, we may develop a better treatment for age-related diseases. Thank you for the previous speakers. So it is much easier for me to introduce some of the backgrounds. So I believe the audience that familiar with there's tons of challenges in the aging research, and one of them, as I mentioned previous, is long life for the human being. So it's a little bit challenging for us to develop any kind of measurement to determine if our intervention work or not. So one of them is chronology age. So it'll be easier to get for every person, but unfortunately this one kind of low biological relevance. So ideally, if you can develop a biological relevant biomarker, biological aging biomarker, then will be more biological relevance. So here's I list several of the current used biological, aging biomarkers. Two of them they are epigenetics biomarkers, and also is transcriptic marker, also is telomere. So all these markers, they have, for example, some of them they are very accurate, but unfortunately, many of them they involve complex experiments. They are cost, and also some of them they are not acceptable. So our work is, can we develop a much simpler or accessible biomarker for aging? So one of that is blood test. As you can see, many of the treatment or many of the patients they are allowing to do this kind of test. If we can develop an aging biomarker from a simple blood test, that would be very benefit to our treatment or to our clinical trials. And we believe, aging is one of the key contributor to the aging-related disease, as I mentioned. So if we know better of the aging, we may develop a better treatment for the aging-related disease. So our company is very interested to apply a deep learning in the drug discovery and also for the aging research. So maybe I can tell you a little bit more about this algorithm. So for the deep learning, they have many algorithms. Here is one of them, it's the deep neural network. So the advantage for this algorithm is, for this algorithm contain normally 3 layers. One called input layer, and output layer, and in the middle there's called a hidden layer. And for the hidden layer, you can have multiple layer within this hidden layer. So the advantage for these algorithms is, for most of our regular algorithms, so we can take only one type of data. For here, we can take different type of data. We can, of course, you can take only one single type of data for, epigenetics data or imaging data or blood test data. But you can also combine these two type or multi-type of data together. Then you can use this one to predict what the outcome. So here I choose age. So because we want to develop aging biomarker, but you can also predict any kind of the health status, or I have an example in the response. We can predict if a person is a smoker or non-smoker. The coming several slides I can show here one of blood test aging clock. So the reason we choose blood test results, as I mentioned, it's cheaper and it's accessible. So everyone is willing to take this kind of test, and so from a simple blood test, there are around 40 parameters. Some of them they are related to the aging. For example, and here I show albumin and cholesterol. So albumin is with aging, with your increase of the age, then the amount of the albumin is decreased, and the cholesterol is increased cholesterol level when we age. But unfortunately, for this kind of simple parameters, we cannot use it as a biomarker. As you can see here, although the trend is albumin is decreased and cholesterol is increased, but by itself is, as you can see, average is very low, and it's impossible to use a single parameters of the blood test as a marker. Here we use a deep neural network algorithm. So we try to combine multiple of the parameters. So here we collect a dataset from Eastern European patients. They contain about 60,000 patient blood test results. So first, after we collect the data, we do normalization of these samples, and we also remove some of the outlier samples due to errors or due to any kind of disease. So after we do this pre-process, then we apply this. Maybe I can start from the simple one. So we use the algorithm. We try to derive the algorithm to predict the patient's gender and also patient's age. So here we first, for the gender prediction, it's much simpler. We can just simply apply all this dataset, train the algorithm, and then we can test the algorithm. We can get a very good result to predict the patient's gender. As you can see, there's only 2 genders, so it's much simpler. But for predict a patient's age, this is a little bit complicated compared to the gender. So we try different algorithms, or we also try different combinations within the deep learning network. So here I just show our final results. The best algorithm we can get the result is we need ensemble about 21 deep neural network. So this is 21 deep neural network. Then we can ensemble the prediction. Then we can predict a patient's age. So here's a plot, patient's real age, and this is patient's predict age using our algorithm. So as you can see, we get very good a trend. So our R-square is more than 0.8. Although our algorithm is not as good as the the epigenetics clocks here, but it's relatively good than compared to the transcriptome clock. But our algorithm have a very advantages. We are very simple and we are very cheap. So every blood test we can just predict the patient's age. See here, I just show some other comparison. We use different machine learning algorithms. So the best result we can get is it ensemble the deep neural network. One of the advantages I mentioned is for use this algorithm, we can identify the top features, or we can identify most important features to predict the age. So here we list the 5 of the top 5 of them. So as you can see, the top 5 they are up by more than 90% of the importance of this prediction. So this 5 is albumin, glucose, alkaline phosphatase, and red cell and urine. And all this is represent the liver function and also lung and heart, etc. So here we are using just one single data set. So we are trying to expand. Can we expand our research to multiple location or multiple data set? So here we acquired 2 new data sets. One from Canadian patients, one from South Korea patients. So here we try to do first we try to do the separation. So we just train, predict, use the single data set, train, predict, use single set. Also, we're trying to consolidate these 3 data sets and do train the algorithm and do the prediction. As a single data set, so we also find in our experiments, so deep neural network can receive the best prediction results from the MAE or from the R-squares. And we also try to cross validation, use different data sets. So you can see if we test a Canadian population and Canadian data set trained algorithm receive the best prediction, and same for the Korea and for the European populations. One of the observation we found is, it looks like South Korea population get a younger prediction compared to the other. So we also try to consolidate all these 3 data sets, because if the biggest data set here we have is about 60,000 samples. So we want to try, can we expand this data set so we combine. After we do the normalization and we consolidate all these 3 data sets and train this algorithm, use the consolidated data, and we also do the same thing to use consolidate this train the algorithm to predict all these populations. Then we get the best data we can get is we compare the single data set trained algorithm, the consolidated data set trained algorithm, we can receive the best prediction result. So here's a summary of the prediction. So as you can see, we this plot predict age compared to the actual age, and we also compare different population, identify the most important key features for these populations, and the 4 of them their common is albumin, glucose, and hemoglobin in the urine. So the three of them were identified same as our previous single data set. So from this, the preliminary results, so we show hey, our algorithms did a good job for to predict a patient's age. And the next couple of slides, we are using the similar algorithm to predict a patient's health status, or called the lifestyle. So here we acquire a data set, different age of patients, and also either smoker or non-smoker. And we do the same thing, normalize the sample and remove any of the outlier of samples, and train the algorithm and predict the patient's age. So here's a prediction results. So here we plot as predict age divided by the actual age and average by log 2. So in that way, if it's if a patient is the number is 0, that means that predict age and the actual age they are the same. So here you can see these two smokers. You can see these three age groups population. So we can always see the smokers predict much older compared to their real age, their real age. So is that that means that at the young age, if you smoke, you may accept your aging. I can skip this one. So here is we see more detail of different age group populations. As you can see, that if young patient, if they are not smoker, they have a very high chance their predict age is much younger compared to their actual age. But if the sample is a smoker, then they have a very less chance to predict as young than their real age, but much higher chance will predict older, either older than their real age. A similar pattern in the 30 to 40 years old group. So you can see non-smokers, they have a very high chance to predict as younger than their real age, but the smokers, they only have a lower chance to predict as younger age compared to their real age, but much higher chance to predict as older than their real age. So we also compare if there's any similar pattern or there's any difference in different patient group who has higher or lower cross level. We divide the patient as higher level and the lower level. We see a similar pattern in these two groups. So we also interested to develop transcriptome agent grok. So here we collect a data set, transcriptome data set. So we normalize this gene expression, and we do two methods. One method use differential gene expression. One method we are own developed called pathway activation genes algorithm to identify the pathway determinants, and use these two type of data to train the algorithm and predict patient's age. Okay, we try multiple machine learning algorithms. So two of them, and right now, two of them, the best result we can get is SVM and DFS. And so you can see the R square is 8.8.8, and so still we get a very good result. And also because we are very interested in drug discovery, so can we apply our algorithm to identify any kind of potential drug target or intervention target? So here we can identify, use different algorithms. We identify the top 20 of the most promising potential features. Then we can translate as a potential drug target. Okay, that's my last slide. So we are interested to develop treatment for the aging-related disease, and so we integrate our different type of algorithm, from the different type of the detection. So we have a platform called the Young.ai. So right now it's in the beta version. So we can track your age based on your based on your photo, based on your blood test, and based on your transcriptome. So this our ongoing project. I'm gonna stop here. I don't have any conclusion.